Good evening, all. I'm Ananya Chatterjee, a student of PhD student of IISc Bangalore. So I am one of two TAs of the course Host Pathogen Interaction or Immunology. So today is the week 5 session. We will discuss. If you have any doubt, you can tell me, or we will start discussing about week 4's assignment. So the first question is, which of the following is not a characteristic property of cytokine? Options are synergy, redundancy, pleiotropy, amplification, cascade induction. So while reading about you know while reading about cytokines, we have heard about synergy, the function synergy, redundancy, pleiotropy, cascade induction of cytokines. But amplification, no, it's not a characteristic of cytokines. So the answer will be amplification. Which of the following is not a function of cytokine? Cell proliferation, immunocells recruitment, cell death inhibition, oxygen transport, inflammation regulation. Obviously, oxygen transport is not a function of cytokine. Cytokine do acts in the scenario of cell proliferation, immune cell recruitment, cell death inhibition, inflammation regulation, okay, but not oxygen transport. If the secreted cytokine has to travel through blood vessels to reach its target and exerts its effect, it is called autocrine, no, paracrine, no, endocrine, yes. So the answer will be endocrine. Okay, which is a true statement about cytokine? So options are they are high molecular weight proteins or glycoproteins secreted by RBCs, long lived, months to years, regulate the intensity and duration of immune response. So yeah, cytokine are not high molecular weight protein or glycoprotein. They are not secreted by RBCs, and also they are short lived. So answer will be regulate the intensity and duration of immune response. Which of the following is a 9 amino acid molecule and plays important function in blood pressure regulation? Thromboxans, bradykinin, insulin, serotonin, prostaglandins. Answer is bradykinin. Which of the following is not a cardinal sign for acute inflammation? So, okay, cardinal sign for acute inflammation. So, which of the following is not a cardinal sign for acute inflammation? Options are redness, rubor, swelling, tumor, pain, dolor, heat, calor, numbness, anesthesia. Obviously, numbness, anesthesia. So, these all four are cardinal sign for acute inflammation. Okay. Ibuprofen and aspirin works by inhibiting the action of superoxide dismutase, inosynthase, cyclooxygenase, pyruvate dehydrogenase, and insulin. Ibuprofen and aspirin works by inhibiting the action of cyclooxygenase. Which among the following properties of cytokines is or are correct? High molecular weight regulatory protein or glycoprotein secreted by WBC in response to intrinsic and extrinsic stimuli. This is correct. Option 3, cytokine's action is long lived, generally ranging from few days to a few weeks. So yeah, option 2 is correct. Which of the following functions of cytokines is or are correct? Development of cellular and humoral immune responses, reduction of the inflammatory response and healing of wounds, control of cellular proliferation and differentiation. So, which of the following functions of cytokines is or are correct? Development of cellular and humoral immune responses, reduction of the inflammatory response and healing of wounds, control of cellular proliferation and differentiation. So, yes, so development of cellular and humoral immune responses is correct. Then, reduction of the inflammatory response and healing of wounds and control of cellular proliferation and differentiation. Yeah, so cytokines do not directly involved in reduction of the inflammatory response, but yeah, they do control the cell proliferation and differentiation. We can check in our. If you have any doubt, let me know, or we can check in the cytokine NPTEL lecture 3. So about the cytokines, these are low molecular weight regulatory protein or glycoprotein secreted by WBCs, low molecular weight regulatory protein in response to intrinsic and extrinsic stimuli. Cytokines action is short-lived, generally ranging from a few hours to a few days. Cytokines regulate the intensity and duration of the immune response. Cytokines exhibits its effect by this kind of actions. So it has a pleiotropy nature. So it means it activates, like IL-4 as a cytokine, it can activate B cell, thymocytes or mast cells for different effect or for different function. Okay, then synergy, that is activated T helper cell, where two cytokine IL-4 and IL-5 both simultaneously act on B cell and induces class switch to IgG. Redundancy, activated T helper cell, where IL-2, IL-4, IL-5 all are like redundantly helping B cell for proliferation. Okay, then we do see antagonism, we do see cascade interaction. Okay, cytokine function, development of cellular and humoral immune responses, induction of the inflammatory response. Yeah, so cytokine induces inflammatory responses, not reduces. Okay, a regulation of hematopoiesis, control of cellular proliferation and differentiation, and the healing of wounds. Yeah, so as I was telling, the healing of wound was correct. Where is the thing? But it's not reduction of inflammatory responses, so partially was correct. Okay. So which of the following statements regarding serotonin are correct? Serotonin regulates inflammation by acting on serotonin receptors that are differentially expressed on non-immune cells. Serotonin acts as a potent chemoattractant, recruiting innate immune cells to site of inflammation. Immune cells have a high capacity to synthesize and release serotonin. Which of the following statements regarding serotonin are correct? Serotonin regulates inflammation by acting on serotonin receptors that are differentially expressed on non-immune cells. No. So, if we go through, yeah. So serotonin is present in hematopoietic stem cells and in high abundance in platelets. Serotonin regulates inflammation and immunity by acting on serotonin receptors that are differentially expressed on immune cells. So yeah, so serotonin receptors are differentially expressed on immune cells, not non-immune cells. So option 1 is incorrect. Serotonin acts as a potent chemoattractant recruiting innate immune cells to sites of infection. This is true, and immune cells have a high capacity to synthesize and release serotonin. This is also true. So option 2 and 3 are correct. So the RNA helicase RIG1 has an essential function in double-stranded RNA-induced innate antiviral responses. So this is a research paper published in Nature Immunology. You can go through the paper, but the take-home message is that the RNA helicase RIG1 has an essential function. It helps in, double-stranded RNA-induced innate antiviral responses. So here the innate antiviral responses is induced by double-stranded RNA, and RNA helicase RIG1 has an essential function on that. Okay. So this is RLR. So these are special like pattern recognition receptors, or example RLR. So different kinds of RLR sensors are there: RIG1, MDF5, LGP2. So where three main domains are present: KERT domain, helicase domain, CTD domain, and TM domain. So KERT domain is present in RIG1 and MDF5. Helicase domain is present in all, and CTD domain is also present. So these are so RIG is retinoic acid inducible gene 1 or DDX58, MDA5 melanoma differentiation associated protein 5 IFIH1, and LGP2 is laboratory of genetics and physiology. Okay. Recognition of RNA viruses by RLRs. So there are different kind of disease: Newcastle disease virus, vesicular stomatitis virus, Sendai virus. So where RIG1 helps. So RIG1 has CAR2 domain, and there are another sensor which is MDA5 in the case of encephalomyocarditis virus. So where these sensors different kinds of secondary like sensor. Example is 5-triphosphate end of RNA, and then they they give activation to IPS1, and from that different signaling such as IRF3/7 or NF-kappa beta gets activated, and type 1 interferons and pro-inflammatory cytokines get produced. So this is a RIG1 domain where you can see the CTD domain, and this is a helicase, and CARD1 and CARD2 domain, and so base pairing is there, you can see. So it's binding with the DNA where there is a triphosphate bond, so where CTD is binding. Okay. So selected viral infection detected by RLRs. So different viral infection which are being detected by RLRs. So Baltimore classification is 1 double-stranded DNA. Virus family example: herpes viridae, herpes simplex virus type 1, Kaposi sarcoma associated herpes virus, Epstein-Barr virus. RLR RIG1 and MDA5, pox viridae, vaccinia virus, adenoviridae, adenovirus. RIG1 and MDA5, RIG1, the double-stranded DNA, two single-stranded DNA. No known examples that activate RLRs. Double-stranded DNA, reoviridae or rotavirus, RIG1 and MDA5. Single-stranded RNA, Piconaviridae, encephalomyocarditis virus, rhinovirus, Coxsackie V virus, RIG1 and MDA5. Flaviviridae, West Nile virus, hepatitis C virus, Zika virus, RIG1 and MDA5. Coronaviridae, SARS coronavirus. Single-stranded RNA, orthomyxovirus, that is influenza virus, paramyxovirus, measles virus, filoviridae, Ebola virus, okay, Marburg virus, single stranded RNA, retroviridae, human immunodeficiency virus, double stranded DNA, heparinaviride, RIG1 and MDA5. Activation of RIG1 by host RNAs during infection. RIG1 immunoprecipitations after HSV1 infection. Highly enriched and particularly RNA 5S rRNA. So that is RNA 5S P141, 5S rRNA pseudogene transcripts host cellular RNAs that are unusual, mislocalized and misprocessed due to disturbances of cellular RNA homeostasis, unmask cellular non-coding RNA including miRNA. Activation of RIG1 by host RNAs in sterile condition. Mutation in RLRs cause spontaneous signaling in the absence of viral infection. So activation of RIG1 by host RNAs in sterile conditions. So adenosine deaminase acting on RNA1, so it's ADAR1 deficiency in humans and mice causes chronic type 1 interferon production and inflammatory pathology. In a breast cancer, the tumor microenvironment results in increased production of the non-coding 5' triple phosphate containing RN7S L1 RNA in stromal cells, followed by transfer of this RNA via exosomes to cancer cells, resulting in RIG1 activation. RLR mediated signaling pathway. So when virus infects us, it comes into inside the cells and then it's releases its genome. So these RNAs get detected by this kind of RIG1 and MDS, and then it activates MAVs. Okay, that is mitochondrial antiviral signaling proteins. MAVs full form. So MAVs, so mitochondrial antiviral signaling protein. So this in downstream activates MAM, which are present in the mitochondria, and then inhibitory kappa beta and then NF kappa beta and the downstream signaling pathway. So this is a RLR pathway regulation, and different proteins and non-coding RNAs are involved. Okay, so some activating post-translational modification and inhibitory translational modifications are also involved here. So okay, so there is some post-translational modification, that is lysine 172 ubiquitinylation. Those are like activating this pathway, and some inhibitory post-translational modifications are also there, which is inhibiting this pathway. RLR mutations and diseases. So there are different genes and where several mutations are associated, and these mutations can do gain of function mutation or loss of function mutations, and there are effects: increased expression of IF1H1, increased expression of IF1H1, disruption of the normal splicing, and so on. And there are different disorders like SLE, AGS, T1D. So RLRs agonist that comprise poly 5' triphospho double-stranded RNA, stem loop RNA 14 and M8 may induce cancer cell apoptosis and immune responses against tumors. Through co-treatment with RLR against high molecular weight poly IC HMW, Scientists have found that the anti-cancer effect of ionizing radiation or IR is enhanced, and the following upregulated FAS expression is responsive to FAS-L induced apoptosis. Therefore, they propose that this is promising to use a combination of RLR agonist IR and FAS-L for treating cancer. SLR14 is a RIG1 agonist which has been observed to induce robust anti-tumor responses is in immunogenic or non-immunogenic tumors and can serve as a powerful immune adjuvant of immune checkpoint blockade therapy. So a nanoparticle delivery of RIG1's natural ligand PPP double-stranded RNA into pancreatic cancer and successfully cease tumor progression. M8, a sequence-modified RIG1 agonist, is a uridine-rich hairpin 5' RNA composed of 99 nucleotides and has recently shown promising potential to induce immunogenic cell death related and damage associated with molecular patterns on tumor cells and synergize with other therapies to stimulate the activation of anti-tumor immune signals. Intratumoral administration of the RIG1 agonist SLR20 can induce immunogenic cell death and breast cancer cell microenvironment modulations, ultimately leading to the inhibition of tumor growth and metastasis. High RIG1 expression is associated with ovarian cancer. A low level of RIG1 suggests poor prognosis in melanoma patients. So again, there are different kinds of genes and mutations associated with it, which leads to gain of function or loss of function mutations. And there are diseases like SLE, AGS, lupus-like nephritis associated with this. RLR ligands in therapeutics. RLRs agonists that comprise poly-IC 5' triphosphate double-stranded RNA, stem loop RNA induce cancer cell apoptosis and anti-tumor immune responses. So, pattern recognition receptors NLR and NLR signaling pathways. In plants, resistance genes plays a critical function against microbial and parasite pathogens, and these genes are NLRs. Therefore, NLRs are first identified in plants. Homologous of the NLRs are present in vertebrates and primitive organisms such as sea urchin. The evolutionary conserved NLRs family play an important function in host defense. In humans, the NLR family is composed of 22 proteins and there are at least 33 NLR genes in mice. So NLR is nucleotide binding oligomerization domain like receptors. Structure and function of NLR ligands. So IE, DAP, Listeria, monocytogenes, chlamydophilia. There are different receptors called NOD1. So induces the production of inflammatory cytokines through NF-kappa beta and MAP kinases pathway. Ligands MDP, Mycobacterium tuberculosis, NOD2 induce the production of inflammatory cytokine through NF-kappa beta and MAP kinases. MDP, lethal toxin of Bacillus anthracis, induces caspase-1 dependent cell death. MDP, microbial RNA, TLR ligand and ATP. Receptor is NLRP3. Induce caspase-1 dependent IL-1 beta and IL-18 production forms. Another P3 inflammasome along with ASC and caspase-1. Legionella pneumophilia control replication of bacteria independent of caspase-1. Okay. NOD1 and NOD2 ligand. So gram-positive bacteria has peptidoglycan. NOD1 and NOD2 ligand is NOD2 ligand is MDP. So there is NAMA and like NUM and NUG glycosylation bond. So where L-alanine, D-glutamic acid, D-alanine are bound, and then these are linked with another glycan chain. So this is intracellular D-gamma glutamyl mesodap (IEDAP) and muramyl dipeptide (MDP), signaling through node 1 and node 2, is ultimately going and then acting on MAP kinase and NF kappa beta. So through TLRs, activation of inflammasome also happens. So this is a signaling pathway where different adapters protein like so there are different adapter proteins which uh, yeah, so there are which give signal to downstream proteins, and then they ultimately acts on certain genes. There's different types of inflammasome, flagellin rod proteins mediated activation, and cytoplasmic DNA. So annular mutation and disease. So there are gain of function mutations and loss of function mutation. So there are different diseases: Muckle-Wells syndrome, familial cold autoinflammatory syndrome, neonatal onset multisystem inflammatory disease, NOT2 and Crohn's disease. So here it's okay. What happens? Reduced NOT2 dependent expression of bacterial alpha dependent in Paneth cell, impaired recognition and clearance of bacteria by intestinal phagocytosis, dysregulation of TLR2 mediated inflammation in intestinal macrophages and or dendritic cells, enhanced production of IL-1 beta. So this is in different annular mutations and different diseases example. Crohn's disease associated with NOT2 mutations in LRR, example R702W. So mice with mutations in NOT2 did not develop gut inflammation but are more susceptible to bacterial and viral infection. So again, there are different proteins which has different mutations and they are associated with certain diseases. If you have any doubt, we can talk about it, and then we'll talk about DNA sensors. Microbes in the human body outnumbers our own cell by option 1 2 times, option 2 4 times, option 3 10 times, option 4 20 times, and option 5 100 times. Microbes in the human body outnumbers our own cell by around 10 times. Okay, option will be 10 times. Which of the following is not a part of innate immunity? TLRs, RLRs, NLRs, these are part of innate immunity. Complement system, part of innate immunity. Then microRNAs, antimicrobial peptides, antibodies. So answer will be antibodies, which is a part of adaptive immunity. Okay, which is a part of adapted or adaptive immunity. Lactoferrin inhibits microbial growth by which mechanism? Endopeptidase activity, ROS and RNS, innate immune cell recruitment, sequestering Fe+2 ions, induce permeability in bacterial membranes. Answer will be sequestering Fe+2 ions. Which of the following is a microbial sensor in Drosophila? We have read about it. It's okay. Microbial sensor in Drosophila. So option 1 TLRs, GNVPs, CD4, RLR and NLR. So we have read that we can see toll-like receptors in Drosophila, but here specifically microbial sensor in Drosophila is given as GNVPs. Let us look into this from our previous lecture slide. So lecture 5. Trying to see that if it's already given in our lecture slides. So the answer is given as GNBPs. Which of the following is microbial sensors in Drosophila? Okay, I will get back to you with this question later. I have to check one thing. So which one is correct? Which one is incorrect? PRR PAM interaction can initiate inflammatory reaction Activate innate immune response, induces type 1 and type 3 interference, initiate a signaling cascade, initiate antibody production. So yeah, PRR and PAM. So this is pathogen recognition receptor and this is pathogen associated molecular pattern. This interaction alone directly can't initiate antibody production. There will be other cascade mechanism, right? There would be different kinds of cascade mechanism, and then induction of B cell, differentiation of B cell, after that only antibody production will be done. So the incorrect answer is this. PAMs are unique to each pathogen, found only in adaptive immunity. No. Responsible for creating antibodies. No. Conserved molecular patterns. Yeah. Conserved molecular patterns on pathogens. Cole's toxin, which was injected in cancer patient, was made up of tumor killing viruses, bacteria and bacterial products, bacteriophages, TNF alpha, complement proteins. It's made up of bacterial and bacterial products. Which among the following statements is correct? Microbes are 10 times more than the number of human cells. Microbiome present in the human body does not include fungi. Every day, an adult human swallow and digest about 1 liter of mucus. So, which among the following statements is not correct? Okay. So, 1 only, 1 and 2, 1 and 3, 2 only. So, option will be 2 only. So, microbes are 10 times more than the number of human cells. This is true. And every day, an adult human swallow and digest about 1 liter of mucus. This is also true. Which of the following statements regarding innate immunity is wrong? Innate immunity evolved 450 million years ago. Since the origin of the sponges, adaptive immune system evolved 700 million years since the first jawed vertebrates. Innate immune genes evolve slowly while pathogens evolve rapidly. Which of the following statements regarding innate immunity is or are wrong? Innate immunity evolved 450 million years ago since the origin of sponges, which is wrong. Adaptive immune system evolved 700 million years ago since first jawed vertebrate. This is also wrong. Innate immune genes evolve slowly while pathogens evolve rapidly. This is true. So option will be 1 and 2. Sorry. 1 and 2. Which of the following statements is or are correct? Lysozyme destroys the cell wall of bacteria. This is true. Dermis gives skin strength and pliability to resist abrasions that could introduce microorganisms. This is also correct. Sebum increases the pH of the skin to a level inhibitor to many bacteria. No, this is not true. Option will be 1 and 2. So today we will start complement system. We'll start complement system. So the term complement refers to a system of factors that occur in normal serum and is activated characteristically by antigen antibody interaction. So you need to understand that the term complement refers to a system of factors. So it's not a single component, but it's a system. That's why it's called complement system of several factors. Okay. that occur in normal serum, and this system gets activated characteristically. So there is some specific mechanisms by which it gets activated by specially antigen antibody interaction. So if you discuss about history, scientist Buchner, German bacteriologist, was the first to observe that the bactericidal effect of serum was destroyed by heating at 55 degrees Celsius for 1 hour. Okay, so he observed that the serum do have bactericidal or killing bacteria activity, but it can be destroyed by heating at 55 degrees Celsius for 1 hour. That means you are destroying some factors which is present in serum. Scientist Pfeiffer, German bacteriologist, discovered the phenomenon of bacteriolysis. Observed that live cholera bacteria could be injected without ill effects into guinea pigs previously immunized against cholera. Okay, so he has observed that live cholera bacteria could be injected without ill effects into guinea pigs which were previously immunized against cholera. So there is something, some factor present inside those animals who were previously immunized against one specific bacteria, and if we again reinfect those, then what is happening? I'm sorry, I just forgot share my screen. Sorry for the inconvenience. Yes. So I was just discussing week 6 assignments. If you have any doubt in understanding week 6 assignment, you can let me know. I will just go through this question number 4 once again after some time, and rest of them I have discussed. So we have today we have started with complement system. So the term complement system, refers to the system of factors that occur in serum. So there is one multiple factors present in the serum that is activated characteristically by antigen antibody interaction. Okay, this kind of system is called complement system, and scientist Buchner, who is a German bacteriologist, was the first one to observe that the bactericidal effect of serum was destroyed by heating at 55 degrees Celsius for 1 hour. So, if you heat serum for around 55 degrees Celsius for 1 hour, then and if you treat bacteria with serum, so if you will be able to see that before heating there is a bactericidal effect, but after heating there is no bactericidal effect. So something is getting destroyed while heating. So scientist Pfeiffer, German bacteriologist, discovered that this phenomenon and discovered one phenomenon and termed it as bacteriolysis. So he observed that if you inject or if you infect one animal who was previously immunized against any virus, be it cholera, be it any not virus, bacteria, be it any bacteria. So he observed this in the case of bacteria. It can be valid if you have. It can be valid for virus if you have good immune antibody generated against it. Yeah, bactericidal effects means so there will be some factors which will help in killing bacteria. Bactericidal means killing bacteria, or it's like bacteria will get burst. Okay, so we can I'll go. Yes, so what happens? He could see that after injecting live cholera bacteria in some guinea pigs who were previously immunized against that disease was didn't get infected. Okay, and plasma from these animals added to live cholera bacteria caused them to become motionless and to lyse. Yes, so this phenomenon is called bactericidal, as I was talking. So means, so their plasma has some factors, okay, has some proteins which you are going to add in live cholera bacteria, and then those factors will induce those bacteria to get lysed or burst or lifeless, okay. This could be inhibited by previously hitting the blood plasma, but this bactericidal property you can inhibit if you hit this plasma before adding. That means you are denaturing the proteins or the factors which are important for bactericidal or killing like bactericidal means to kill bacteria. This activity is needed, so if you are destroying those, then you won't be able to see this bactericidal effect. So scientist Pfeiffer called this as bacteriolysis, and it became known as a Pfeiffer's phenomenon. So this is important to know what is Pfeiffer's phenomenon. Bordet established that immune bacteriolysis and hemolysis require two factors. One is a heat stable antibody, and another factor is a heat labile factor alexin. Paul Ehrlich coined the term complement, replacing the term alexin, because this factor complemented the action of antibody. So here comes the story of complement system. Complement means complementary, okay. So it's get activated in the presence of antigen antibody complex, and then it also fastens the process. So there are two main thing is required to have bacteriolysis and hemolysis property. One is heat stable antibody, another one is heat labile factor alexin. This is further in future it was termed as complement or complement system. It is named complement system because it was first identified as a heat labile component of serum that complemented antibodies in the killing of bacteria. Consists of serum and cell surface proteins involved in defense against pathogens and tissue damage mediated by antibodies. Consists of serum and cell surface proteins involved in defense system involves in defense against pathogen and tissue damage mediated by antibodies. The complement system is a major effector of humoral branch of immune system. Plays major role in both innate and adaptive immunity. So what is the general properties of this complement system? These are non-specific. This consists of 5% normal serum protein destroyed in 30 minutes at 56 degrees Celsius, that is heat labile. Bind only to antibodies that have combined with their antigens. Only immunoglobulin M, immunoglobulin G, 312 fixed complement. Binds to FC portion of immunoglobulin. 30 proteins, C components per plus properdine plus control proteins. 9 different fractions C1 to C9. Complement is a complex of 9 different fractions C1 to C9. Fraction C1 occurs as calcium plus 2 ion dependent complex, which on chelation with EDTA yields 3 protein subunits C1, Q, R and S. Fractions are named C1 to N in the sequence of cascading reaction, except C4 comes before C2. So when a fraction acquires in enzymatic or biological activity, it is indicated by a bar over the component number. That is, if C1 gets activated, then C1 bar. They are mainly synthesized by hepatocytes, also produced by blood monocytes, tissue macrophages and epithelial cells of the gastrointestinal and gastrourinary tract. Complement proteins made as zymogens, activation by cleavage. Example, C4. So here you can see that from C4A, which is a smaller fragment, gets so activated by cleavage and forms C4b, which is larger fragment. Exception C2. C2A is larger fragment and C2b is smaller fragment. Complement activation. So normally, yes, tell me. Yes, yes. So zymogens means, you know, those are enzymes, but it needs some cleavage activity. So this is a protein, okay, and so this is a zymozyme, but it's not active now. It's inactivated. So it needs to uh, break some bonds and then form two different fragments, and then it becomes like active enzymes. So here C4 is zymozymes, and then some cleavage is happening and it's forming C4A and C4B. So and it's becoming active enzyme, like which will do the function further in cascade mechanism. Yes, yes. So yes. So like cleavage is needed for their activation. I can show you some more example. Just a second. Mm. Yes, so if you see in Google, I will share the screen. Mm, Wait, my internet is poor today. Yeah, so if you just Google zymogens, you can find one image. Internet is super slow today. Anyways, yeah. So you can see this is a this is a protein, okay, or you can say enzyme, which is not active. So there are different environmental factors which are coming in the play in the action, and then either it is getting clipped or some autocatalysis activation is happening, okay, maybe like, so this is a protein, so some part of the protein is activated, somehow some bond formation or somehow something is happening inside. This is called autocatalysis. And if not autocatalysis, it can also uh, gets clipped into two substance, okay. So here you can see, so breaking a bond also can be called can be called, this autocatalysis activation. So here you can see this was zymogen, but after breaking a part of it apart, it is becoming active enzyme. So this is it. So complement activation, not. Yes, yes, my internet is very bad today. I'm connecting with my cellular network. I'm not connected with Wi-Fi. I am sure. So I am recording the video where you will find the voice very clear, because I'm screen recording my screen and audio as well. So I'm uploading in my YouTube channel, which is already the link is shared with you all. NPTEL lectures by Ananya. You can, if you want to just revisit, you can. I am writing it in the chat section. So I will start again. Complement activation, normally present in inactive form in body. So these complement systems or the proteins associated complement system are normally inactive. Activity is induced by antigen antibody combination. Components react in a specific sequence as a cascade. Cascade is a series of reactions in which the preceding components acts as enzymes for the succeeding components. Components are clipped in dissimilar fragments. Larger fragments join the cascade and the smaller fragments released in the medium possess some biological functions. Complement activation, lysis of cells and bacteria, promotes virus neutralization, opsonization, immune clearance, amplification of inflammatory process by increasing vascular permeability, release of histamine from mast cells. So one example, number 1, lyse bacteria by forming MAC. So complement system can lyse bacteria. Okay. Function 2, tag pathogens to enhance recognition. So some proteins associated with complement system can get attached with some specific bacteria to tag those bacteria and to enhance the recognition of those bacteria by phagocytic cells and distraction by phagocytes, which is called opsonization. Function 3, activate inflammatory response by triggering release of histamine from mast cells. So activation of inflammatory response, it can activate inflammatory response, so it triggers release of histamine. So basically complement systems interacts with complement receptors, and then some cascade mechanisms, it also interacts receptors present in mast cells, and then some different signaling pathways, some cascade receptors, cascade mechanism, it releases histamines or inflammatory molecules from mast cells. Function 4, enhance clearance of antigen antibody complexes. So suppose there are antigens which are getting presented by antigen presenting cells, and then antibodies are attached with it. Okay, so complement system comes into the action, and then it increases the clearance process. Basically it fasters the process of antigen antibody complex clearance. Complement activation pathways, so there are different pathways, we are just describing what it can do, but there are different pathways or signaling process, you can say that. Because why signaling? Because it's a cascade mechanism. One protein it's activating another protein, that protein is activating another substance, like that. So that's why it's called a cascade pathway. Or so we'll discuss complement activation pathways. C cascade is triggered off by 3 parallel but independent pathways. So complement pathways, also called C cascade pathway, differ only in initial steps. Once C3 activation occurs, the subsequent steps are common. 3 major pathways of complement activation. Classical pathway, activated by antibody bound to antigen. Alternative pathway, activated on microbial cell surfaces in the absence of antibody. Lectin pathway, activated by plasma lectin that binds to mannose residues on microbes. Central event in complement activation is proteolysis of C3 to generate biologically active products and subsequent covalent attachment of C3b to microbial cell surfaces or to antibody bound to antigen. So these are 3 basic pathway: classical, alternative and lectin. We have already discussed Pfeiffer phenomenon, and the complement system we have discussed about Bordet principle, and then lytic activity, bactericidal activity, Paul Ehrlich also conducted the similar experiment, coined the term complement. So, complement term was coined by Paul Ehrlich. Components of complement, if you see, so this is the soluble proteins or glycoproteins. Glycoproteins means glycosylations happens in those proteins, so glycosylated residues are there or glycans are present in the protein, synthesized mainly by liver hepatocytes and other cell types. So these proteins are synthesized mainly by liver cells, or also called hepatocytes, or other cell types. And we have discussed that 5% of serum globulins are complement proteins, and we also discussed that it circulates as inactive proenzyme, which is also called zymogens. So proteolytic cleavage happens and it removes one inhibitory fragment and exposes its active site. So it can do lysis, we have talked about this. It can do opsonization and also activation of inflammatory response. Nomenclature of complement designated by, so complement nomenclature we also, yes, tell me. Opsonization is, so you have heard about phagocytosis. So opsonization is, yes. So opsonization means just, the enhancing the phagocytosis process further. So complement systems proteins are tagging those bacteria, and then bacteria in further is getting phagocytosed. Okay, so this is the same, yeah, the same part, but it's some other proteins are associated in helping the process. Can show you some picture of it if my internet works. So it's not like mandatory that only opsonization happens if complement system proteins helps, but some other factors also can get associated just to foster the thing. So if we see, opsonization can be because of different other factors. I will talk about it, but complement protein mediated opsonization is that you have to go in details that there are different factors, and by cascade. Oh, so okay, we'll talk about it later, because I have not introduced to you yet, like C3b, C4b, these things are not introduced yet. But if you see opsonization in overview, so there are several research paper you can go through which are very much interesting. So pathogen opsonization, there is a video in YouTube you can see by complements how it. I'll just show you if it plays. It will be interesting. See if it's visible. So see, phagocytosis of pathogen opsonized by complement system. So basically, one phagocytic cell can phagocytose different pathogens. But if those pathogens are previously recognized by complement system, and those complement system help in recognizing the pathogen further to those phagocytosed cells, So this process gets enhanced. So here it's happening that phagocytosis of pathogen opsonized by complement. So opsonized means it's getting engulfed by complement system. So this is one example of mannose binding lectin pathway. This is alternative pathway. Here it would be classic pathway. See, antibodies are, so pathogens are getting recognized by antibody, okay, and then it gets engulfed. These are different antimicrobial peptides. It's just breaking the pathogen. So yeah. So this is called opsonization. Nomenclature. Oh, something gets activation of innate cells. You can see this video if the video is running. So it's showing that it's showing that activation of innate cells against virus infected. Oh, sorry. So another video got played at its own. You can see the video. Activation of innate cells against virus infected cells. So this is an infected cell and this is natural killer cell or NK cells. Yeah, it's nice quick videos. Yeah, thanks. So complements are classified into 7 functional categories. Functional protein classes in the complement system, binding to antigen antibody complexes and pathogen surfaces. Example C1q, binding to carbohydrates structures such as mannose or N-acetyl glucosamine on microbial surfaces, which are mannose binding lectins, phycolins, properdin. Properdin is also called factor P. Activating enzymes C1R, C1S, C2A, and so on. Surface binding proteins and opsonins, example C4b, C3b. Yeah. So examples of, if you are asked example of some complement proteins which help in opsonization, your answer will be C4b, C3b. Peptide mediators of inflammation C5a, C3a, C4a. Membrane attack proteins or MAP, C5b, C6, C7, C8, C9. Complement receptor CR1, CR2, CR3, CR4. Complement regulatory proteins, there are a lot of names, C1 INH, DAF, CD59, and etc. So one example of alternative pathway, C3 protein undergoes spontaneous hydrolysis to C3H2O to initiate eventual deposition of C3 convertase on microbial surfaces. Classic pathway, C1q interacts with pathogen surface or with antibodies bound to the antigen surface. Lectin pathway, mannose binding lectins and phycolins recognize and bind carbohydrates on pathogen surface. So there are different mannose binding lectins. It can recognize mannoses or glucose. You know, there are different viruses, different pathogens which has glycosylated protein. So they in their cases, glucose or mannose residues are exposed. So those residues they target, like those complement system target, like those are lectin proteins. So they have affinity towards glucose or mannose. So they bind to it and then recognize those antigens. So three pathways: lectin pathway, classical pathway, alternative pathway. In all this you can see that the cascade mechanism is activating different other complement proteins, example C4 C2, here also C4 C2 C3 in alternate pathway, and all pathways generate a C3 convertase which cleaves C3, leaving C3b bound to the microbial surface and releasing C3a. So what is happening, C3 convertase cleaves C3 and produces C3b and C3a. C3b remains bound to the microbial surface, but C3a, the other fragment, gets released. And then C3a and C5a recruit phagocytic cells to the site of infection and promote inflammation. This can be done. Then phagocytes with receptors for C3b engulf. So where you can see C3b gets remains attached with a microbe. So then phagocytes with receptors for C3b, there are cells which has receptor for C3b. They engulf those pathogens associated with C3b and then they degrade or destroy the pathogen. Completion of the complement cascade leads to formation of membrane attack complex or MAC, which disrupts cell membrane and causes cell lysis. This is the final event. So this is classical pathway. So here you will be able to see that different complement proteins are getting activated in cascade mechanism. So this is classical pathway, which is helped by antigen antibody complex. This is lectin pathway, involved mannose binding lectins, and this is alternative pathway. Ultimately they are all forming membrane attack complex. Complement classical pathway, antibody dependent, activated by antigen antibody complex, most commonly immunoglobulin M and immunoglobulin G. Early stages involve C1, C2, C3 and C4. C1 of classical pathway, C1 macromolecular complex, C1QR2S2, two molecules of C1R and C1S and one molecule of C1Q. This is the structure of the protein or macromolecular complex. They have a stalk region and has head region. So here C1R and C1S, this two molecules, this is C1Q molecule. The total in the whole macromolecule is called C1 macromolecule. So this is the antibody structure and function. It's a Y-shaped. Here you have heavy chain, you have light chain. Heavy chain and light chain are getting attached by disulfide bond, and then two heavy chain as well are getting bonded by disulfide bond. So it has antigen binding domain and one FC region or constant region. So this has effector activity or different biological activity. So there are different antibody types: IgA, IgG, IgT, IgE, IgM. IgM is pentameric. IgA can be dimerized by J chain. This is a J chain. And this is IgG, IgD, and this is IgE. Yes, so this dimerization is required. So yeah, obviously if there is J chain, there is required. So sometime what happens, if the antibody is dimer, okay, and J chain is making the dimeric bond, it's just in interaction of two monomers is happening. So if there is a macromolecular complex at it interacts, IgA is interacting, so J chain is giving a flexibility so that this is one IgA, this is another IgA, is binding like this. So it's a weak macromolecular complex and it's trying to find those epitopes in a very flexible manner. Okay, so yes, so this gives, yes, yes, yes. And in IgM, those are pentavalent. Pentavalent means, if like you can see this five epitopes, like five binding motifs you can find, and there also one J chain is present, and those five pentamers are linked by disulfide bonds. So this is complement activation. Mm, okay, I will just see. So there is a science static paper. It's showing that the J chains are not necessary for formation of IgA dimers. However, they're essential for binding polymeric IgA to secretory component and are required for the efficient transportation of serum IgA into bile. So it gives solubility as well. So what is the role of J chain in secretory immunoglobulin formation? So the joining chain, which is joining two monomers, as is a small peptide expressed by mucosal and granular plasma cells, which regulates polymer formation, as I was telling, forming dimerization. But there is also role in making it secretory or solubility. So how many J chains are there? There are, although it's generally believed that pentameric IgM and dimeric IgA contain one J chain per polymeric molecule, some research group have reported that two J chains for IgA and three or four J chains for IgM. So it's conventionally one for each, but in some research paper you can find that they have reported two J chains for IgA. What is secretory? So if you are asked that what is the secretory component for IgA, secretory component is a proteolytic cleavage product of the polymeric immunoglobulin receptor, which remains associated with dimeric IgA in seromucous secretions. Polymeric IgA binds to the polymeric immunoglobulin receptors on the basolateral surface. Okay, there's nothing is said here. Mm, there are different papers. Okay, okay, yeah. So you can ask questions, so that will give me more interest. Yeah, thank you for asking. So there are different papers you can read, and if you have any questions, we can discuss more. So there are one paper, structural requirements for incorporation of J chain into human IgM and IgA. So this is complement system, the cascade pathway in diagrammatic view. So what is happening here? C1q binds with antigen bound antibody. Okay. Then the C1q is the whole system, and then what happens? C1r activates autocatalytically and activates the second C1r. Both activate C1s. Okay. C1s cleaves C4 and C2, C4a and C4b, and in C2, C2b and C2a, C4 binds the surface near C1 and C2 binds C4, forming C3 convertase. Okay. This is C3 convertase, which is also called C4b C2a. C4b C2a, or in short C4b 2a. So this C3 convertase hydrolyzes many C3 molecules. Some combine with C3 convertase to form C5 convertase. So this C3 convertase can hydrolyze C3 into C3b and C3a, and also it can form C5 convertase, which is C4b 2a and 3b. The C3b component of C5 convertase binds C5, permitting C4b2a to cleave C5. Okay, so this is C5 convertase, which was C4b2a and 3b, and they binds to C5, which is basically C5b plus C5a, and then it's like which is cleaving C5 into this fragment. C5b binds to C6, initiating the formation of membrane attack complex. So this is C5b, from here C5b, C6, C7, and also it gets associated with C8 all together, like C5, C6, C7, C8 all together binds to C9 in the present in the membrane, and forming membrane attack complex. So this is one. Sorry? Yes. So the whole thing you don't have to memorize, but if you read again and again, so there is one basic flowchart. You don't need to remember this C1q R2 S2. It's complicated. I have also forgotten. So you just have to remember that what is like. So there are some important steps. That is, you need to remember what is cleaving C3. So C4b C2a. You once you read one, so you need to see some videos, diagnostic videos from where these pictures has been taken. You will get so once you understand, it will be uptaken by your brain. So what happens that after C1, it's going to it cleaves C4 and C2. This much is fine, but you don't need to remember that C1 activates C1r and C1s. This is not required. This much specific question you won't get asked, and also it's not relatable. Where once you get question, you can just read some paper and then have a overview. So and then this is important. So what is C3 convertase? You can remember that it's made up of C4b and C2a. So C is not mentioned twice, but it is C4b plus C2a. So like this, some important steps like C5 convertase, and then after that, after C5, you can see C5 is getting associated with C6, C7, C8 all together, and then C9 is present in the membrane. Already it's tagging the antigen, so all together is coming here and then forming a membrane attack complex. So this is the story. So some membrane attack complex you can see under electron microscope. So it basically forms a pore in cell membrane where ions and small molecule can freely pass through the pores, and if pore is getting generated in the cell won't be able to maintain osmotic stability. This is how it kills the target. So there are alternative complement pathway which is antibody independent. Component of innate immune system, early stages involve C3, factor B, factor D, properdin, initiated by cell surface constituent foreign to host, for example gram and gram positive bacteria. So initiators of the alternative pathway of complement system, you no need to recall all those 10 names. So there are pathogens and particles to microbial origin. There are different pathogens, different gram negative and gram positive bacteria. Non-pathogens, that is human IgE, IgG in complexes. So these are initiators of alternative pathway. In alternative pathway also you can see C3. Basically the complement system, you have to remember that the protein gets these are in zymogens form, that is inactive enzyme form, and get cleaved and then becoming active enzyme. So C3 is becoming C3b and C3a, and then again the same pathway it's coming. So the final thing is the same for every complement system, you can see, but the initiator is different. Okay. So we have talked about complement lectin pathway as well, where initiators are mannose and lectin interaction. This is one video of complement system you can see in YouTube. Regulation of complement. Complement components are capable of attacking host cells. Complement components undergo spontaneous inactivation if they are not stabilized with other components as its highly labile components. C3 convertase is a major amplification step in all three pathways. So this C3 convertase you have to know, you have to remember, because it's common in all three pathways, therefore that needs regulation. Soluble factors regulating complement. So yeah, this much you don't no need to recall all these things, but some important thing is that here protein S ligand or binding factor is C5b 6 and 7 complex, and action is it inhibits MAC formation. MAC means membrane attacking complex formation, it inhibits protein S. And then again protectin, it inhibits MAC formation. So microbial pathogen invading complement system. So yeah, so there is system, there will be police system, but there will be some invasion by invaders. So there are different pathogens, and there are evasion molecules associated with them. They target some host factors and their mechanism of actions are there. It's very difficult to remember all these things, so just few example for you guys. Complement deficiencies. Okay, so deficiency of C1q, C1r, C1s, which is all are part of C1. C4 and C2 show enhanced immune complex and show SLE like symptom. Glomerulonephritis, vascularitis, these are different diseases where we can see deficiency of our complement system. Individual also show recurrent infection by pyogenic or pus forming bacteria. Deficiency of factor D and properdin show Neisseria meningitis infection but not with meningococcal bacteria. Okay. So deficiency of mannose binding lectins. Yes, tell me. Hello. Yes, yes. It's disease. So SLE means autoimmune disease. Okay, where it deficiency of this complement system enhance some immune complex, where immune cells detect our own cells and get killed. Okay, so SLE like symptom. SLE is a autoimmune disease. Yes, so you can Google and read about it more. Yeah. So also deficiency of mannose binding lectin show increase in respiratory tract infection. Deficiency of C3 show frequent severe bacterial infection. Protectin deficiency can be of high risk of thrombosis. Anti-C5 is used for the treatment of paroxysmal nocturnal hemoglobinuria, that's a big name, and come in the name of ecluzizumab. Deficiency of C1 cause hereditary angioedema. Okay. So adaptive immunity introduction. So we will introduce adaptive immunity. So how different arms of immune system are linking. So there are cell components which can be non-immune cells or immune cells. They have pattern recognition receptors. Those will detect pathogen associated molecular patterns which are present in antigen. They will initiate inflammation, they will induce type 1 and type 3 interference and activate innate immune responses, and altogether initiate the pathogen specific adaptive immunity via B and T cell. Key cellular players of immunity. So the part of innate immune cells are neutrophils, eosinophils, basophils, NK cells, also dendritic cells. Adaptive immune cells are B cells, T cells, and there are some cells which acts very crucially in bridging innate and adaptive immunity. You can say those are innate immunity. Also, you can say they helps a lot as activating adaptive immune cells. Those are macrophages and dendritic cells. Comparison of innate and adaptive immunity. So innate immunity is already present in the body. Adaptive immunity you need to get developed throughout your survival. Innate immunity broad specificity, adaptive highly specific, fights with any living or non-living foreign entities, but adaptive develop highly specific immune response if not cleared by innate immunity. Innate is very rapid, adaptive immunity is slow. Innate is limited and lower potency, adaptive immunity is high potency. Time span every time the response is generated irrespective to the number of exposure. Adaptive, once immune response developed, the immunity can be lifelong or short, depends on pathogen. Innate immunity is generally inherited from parents and passed to offspring, but adaptive is not passed from parents to offspring, hence it cannot be inherited. Innate cannot react with equal potency upon repeated exposure to the same pathogen. Adaptive system can remember the specific pathogen which have encountered before. Innate present at birth, adaptive develops during a person's lifetime and can be short-lived. Innate none, adaptive immediate and delay hypersensitivity. Innate, there is no memory cells associated with innate, but adaptive immunity, there is long-term memory. Innate immunity diversity is limited, adaptive immunity diversity is high. Innate immunity, alternative and lectin pathways, complement system activation, adaptive immunity, classical pathway. Innate immune cells present in skin, mucous membrane, temperature, pH. Adaptive immunity, lymph nodes, spleen. Composition is composed of physical and chemical barriers, phagocytic leukocytes. Adaptive is composed of B cells and T cells. Innate immunity development, evolutionary, older, and is found in both vertebrates and invertebrates. Adaptive immunity system has been developed recently and is found only in the vertebrates. Example, WBC is fighting bacteria causing redness and swelling when you have a cut. Chickenpox vaccination, so you won't get chickenpox disease because your immunity, there is generation of adaptive immunity and they are going to generate memory cells which will remember the foreign body in future infection. Yes, tell me. Divergence. Okay. Yes. So in innate immunity, there are specific cells which will act as classical pathways. But adaptive immunity, if I get infected with the same virus and you are also getting infected with same virus, there will be different kinds of antibody will get generated. So antibodies also you have seen that there are different subtypes. So that means the diversity, the adaptive immunity is how you are going you are going to adapt it and how I am going to get adapted. So this is diversity difference. So there are different antibody will get generated. So they are also switching, class switching will happen. So diversified antibodies, we'll discuss in next class that how class switching happens in different antibodies. So this is called diversity. And limited means, yeah, limited means there are some limited cells will act in classical ways. So this is characteristic of B cell and T cell. B cell involves binary complex of membrane, Ig, immunoglobulin and antigen. Binding to soluble antigen, yes. Involvement of MSC molecule, no. B cell chemical nature of antigens can be protein, polysaccharide, lipid, epitope properties, accessible, hydrophilic, mobile peptides containing sequential and non-sequential amino acid. For T cell, we'll discuss this more. We'll discuss this factor in tomorrow's, like next day's class, in more detail. T cell involves ternary complex of T cell receptor, antigen and MSC molecule. Binding of soluble antigen, no, there is no binding with T cell. Requirement required to display processed antigen, mostly proteins but some lipids, and so on. What is the difference between immunogenicity and antigenicity? Immunogenicity induce immune response such as antibody production. Antigenicity not necessarily induce immune response but can induce immune response if appropriately modified. Hapten is too small, lack immunogenicity. These are hapten molecules. If you add some carrier molecule, the size is getting bigger. So if hapten is coupled with some carrier molecule, immune response can be induced against this haptens. This is called hapten carrier conjugate. Produces three types of antigenic determinants: antibodies to hapten, antibodies to carrier, antibodies to hapten carrier conjugate. So we will discuss about adaptive immunity antigen in our next coming class, next Tuesday. If you have any other question, we can discuss today. So Shilpa, you are in bachelor's, master's? Nice. Where are you doing your master? Okay, okay, nice. So you are planning to come in research field or planning to do PhD? You will listen. Mm-hmm. So yeah, yes, do well in your master's dissertation. You will be doing in your um, parent college, or you can do outside in different institutes. Okay. Okay, okay. Which question? Yeah, yeah. Tell me. Week 6 means is already due date. Okay, due date is there. Yeah, tell me. Ye- yes, yes, yes. Both you can do. You can share screen as well. Yes. Yes, yes. Tell me. Mm-hmm. Okay. Okay, okay, okay. Yeah, yeah, yes, yes, yes. So agonist means which helps, and antagonist means which doesn't help. Okay. So we have to go through those information slides where I will see. It was there. The exact information I can remember, but if we, I can make you understand if we see the slide. The question what was the SLR? Okay, I'm just seeing. Hmm. So, if you, so you have found in the lecture slides about um, this terms. So yeah, so it's there in, yeah. So SLR14 is a RIG1 agonist, okay, which has been observed to induce robust anti-tumor responses in immunogenic or non-immunogenic tumors and can serve as a powerful immune adjuvant. Okay. So the question was, what was the question? Please repeat. SLR14 is a SLR14 is a, you can, okay, okay. Yeah, got got it. It will be agonist. Okay, it will be agonist of RIG1, but antagonist of what? Okay. Mm, hmm. If the question is correct, then we have to think that which is the antagonist of RIG1, then that will be the answer. And if there might be that there is just, they have written it antagonist but it will be agonist, there is chance of that also. Yeah. Yeah, tell me. uh rig 1 also is yeah. Oh, so rig 1 MDA5 AM2 LGB2 TLR7 acts as a powerful immune adjuvant and induces anti-tumor response, is an agonist. I think it should be agonist. It should be this much straightforward. Hmm. Yeah. Yeah. Yes, yes, yes. Because they won't ask this much complex question. the It acts as like it helps rig one, but yeah, it should be agonist. You can mark rig one. Yeah. Yes, yes, it's okay. Yeah, it should be agonist only. It's okay. Thank you. Bye bye. Good evening.